Understanding bioenergetics and metabolism. Today, we will look into the concepts, the underlying principles, and also the examples with respect to metabolic pathways, which is very important from CSIR NET Life Science point of view. But let me make sure for all the attendees and viewers today that this topic is a very basic concept. And if you are preparing for any life science PhD entrance exam, then definitely you have to watch this video because it will help you in many other concepts as well. First of all, the basic question, what is bioenergetics? Well, we know the characteristic of living organism is metabolism and reproduction. We have studied from childhood, and we need energy for the work, to do the work in order to survive and also reproduce. That is why in biological systems, we have something which is called as energy transduction. We know energy can't be created nor be destroyed according to the first law of thermodynamics, but it can be transferred from one form to the other form. So that's why the potential energy, chemical energy, kinetic energy, these all convert into each other, and that is where the entire energy to do work in biological system comes from. Bioenergetics is the quantitative study of cellular energy transduction, how the cell harvests the energy from the food that we consume, how it breaks down to get the energy currency in order to perform the cellular functions, also the nature of function of the chemical processes which are necessary for the energy transductions. This is what is studied generally in bioenergetics. Obviously, it is involved and linked with thermodynamics. You must know the fundamental laws of thermodynamics, which I just said. The first law, which is in energy conservation. Energy can only be transformed from one form to the other. And the second law is increase in the entropy. Well, what does entropy mean? It means disorder. So, if molecules are not present in an ordered form, but it is present in a random form, then we call that it is a disordered manner of representation. And if you have increase in the disorder, that means we have the increase in entropy. So entropy is a measure of the disorder. So second law says that whenever you have any molecules, then always they will have a tendency to increase the entropy. I will give you a funny analogy for that. You people must be cleaning or organizing your room, right? But after some time, you will see that in course of time, by itself, the room becomes automatically disordered, isn't it? So the things are not in place anymore. So that's how you can remember that the biological system is also same. They like to be more free. So there will always be a tendency to increase the entropy. There will always be a tendency to create disorder. Okay. Now coming to energy, yes, energy it is required by all organisms. Basic classification of energy, you have kinetic energy, which is nothing but energy of motion. Examples you can take as heat and light energy, which will give the molecules more energy, isn't it? If you provide heat to this molecule, let's say, right, so it will start to vibrate, right? So that is the energy which is giving it the motion. That's why it is an example of kinetic energy. Potential energy, it is something which is the energy of position. Means the atoms, they have something, atoms, bonds, molecules. They have something which is called as internal energy, which includes all the energy stored in its bonds in the form of if you provide work to it or if you provide heat to it. So these are all providing more energy to it. Totally, whatever energy it will have, it is internal energy. It includes the energy stored in the chemical bonds, right? Now, a very important one, two types of energy reactions. One is endergonic reactions, another one is exergonic reactions. So what is endergonic? See, first of all, whenever this word gonic comes, we are talking of a thermodynamic term which is called as free energy. Since Gibbs was the person who have given the term, it is also called as Gibbs free energy. Why it is called as free energy? Simple, it comes for free. Okay, so if a cell or a molecule, it can release some amount of energy from its molecule, okay, without having to compensate for the energy. That means it can just give away. It doesn't have to come back to it, okay. Similarly, any molecule can just accept the free energy. So that's why it is called as free energy. Some amount of energy can be either released from a molecule or it can be acquired by a molecule. Okay, so chemical reaction that requires endergonic reaction, which requires a net input of energy, means let's say you have these carbon dioxide and water which is reacting in photosynthesis. Well, we will call them as reactants, and the glucose and the carbon dioxide and the oxygen that it produces, these are called as the products. Okay. Now, if these reactants have a less free energy, okay, so we will say that let's say G1, it is less for the reactants, and the free energy for the products, it is more. So as you can see, in order to go from reactant to product, this is the forward direction, you will have to gain some amount of energy, isn't it? Then only the energy of the product will be increased. So such kind of reactions are what are called as endergonic reactions, which will have to require a net input of energy. For example, as I told you in photosynthesis. Now, if we have to find the change of free energy, this delta term we write to represent symbol to represent energy. Delta G over here, we have to always take from the final minus the initial. Now, initial here is less and final here is more. So, if you subtract a lower value from a higher value, the value, that answer you're going to get will be in positive. So, very, very important point for endergonic reactions, the change of free energy for the reaction to take place will always be positive. What about exergonic reactions? These are the chemical reactions that release energy. For example, if you take respiration, here it is the other way around. Cellular respiration, where the reactants have high free energy, okay, and the products, they have low free energy. Now, a very important concept I'm going to tell you, do not forget it for the rest of your life if you are going to continue in life science career, that the molecules which have low energy, okay, anything which has lower energy, they are going to be more stable. Okay, so any process which gets to lower their energy, they are going to be favorable processes. Okay, so as I said regarding entropy, that the tendency is always to increase the disorderedness or entropy. For free energy, it is the other way around. The tendency is always to decrease the energy. Okay, so that's why the processes in which the energy will be lowered, those processes will be called as spontaneous or favorable processes. So here, if we have to calculate the delta G, then again we have to take G2 minus G1, but here we are subtracting a higher value from a lower value. So what will be the result? It is going to be negative. Very, very important concept, that in exergonic reactions, these are the reactions that will release the energy, and here the delta G is going to be negative. Okay. So here is the graphical representation for the same. In the y-axis, you have Gibbs free energy, and this is in course of the reaction, the time taken for the reaction. Initially, at the beginning of time, obviously they are present in the form of reactants, where in endergonic reactions, as I said, the reactants ground state. So this is where the Gibbs free energy point is. As you can see, it is less. Okay, so Gibbs free energy is less over here. But products Gibbs free energy, if you can see, it is more, it is higher. So obviously, if this reaction has to take place, you will need more input of energy. Okay, and if you have to increase energy, that means this reaction is not spontaneous. Okay, energy is absorbed here, and that's why the delta G is more than 0. Coming to exergonic reactions, when you have the free energy of the reactant, it is very high, and free energy of the product, it is less. In this case, it's a favorable reaction. This reaction will happen on its own without the input of any other energy. So such processes are generally irreversible. Irreversible means by its own, you cannot reverse this process until and unless you provide energy, right? So the flux or flow of these kind of reactions will be always towards the product, since the tendency is to lower the energy. You can also say it in a different way, that the equilibrium of this reactants is always in the forward direction or towards the formation of the products or towards the right hand side of the reaction. And as I already told you, delta G will be less than 0. That means it will be negative. Moving on, coming to metabolism. Metabolism is a combination of two processes. One are catabolic processes, another are anabolic pathways. So catabolic reactions, these are metabolic reactions which release energy. Okay, that means we just saw they are exergonic in nature, they can release energy, in which complex organisms, complex molecules is broken down into simpler compounds. For example, if we are consuming a piece of bread, then we are intaking carbohydrate. If we are taking an egg, we are consuming protein. With butter, we are consuming fats. So when we take bread and butter and egg for our breakfast, then we are actually getting the fuel to generate energy from it. Now what will our cells do? From the digestive system, it will be absorbed to the intestine into the bloodstream, it will reach the cells, and in the cells this catabolic reactions will be taking place to break down this complex compounds. Actually it doesn't go in the form of complex carbohydrate, it will go in the form of monomers like glucose, fructose, galactose, like that. Okay, and those glucose molecules then will be broken down into further smaller molecules. Ultimately it will be giving rise to energy through a process of oxidative phosphorylation, which we can discuss in another video. But the idea is that where a complex molecule is broken down into carbon dioxide, water, ammonia. As you can see, these are very simple precursor molecules. And these pathways, actually these processes will be consisting of more than one reaction. So that is why we will be calling it as pathway, or it can be cyclic also, as in the example of Krebs cycle. These pathways will consist many reactions, but overall when I say they release energy, I mean it is the net result. So let's say in case of glycolysis, there are total 10 reactions. Does all the 10 reactions of glycolysis release energy? Obviously no. So that's what is meant by catabolic. If you see, in the first 5 reactions of glycolysis, we need energy. So 2 ATP is required. But in the next 5 reactions of glycolysis, we will see that the 4 ATP is produced. So what is the net result of that? Ultimately 2 ATP will be released. Okay. So that's how we calculate the net result of catabolic pathways. That's how you will understand whether it is a catabolic reaction or an anabolic reaction. So example of this is cellular respiration, as I said, leads to oxidative phosphorylation to generate energy. That's why from catabolic reactions, as you can see, have generated energy. Now this energy or the ATP will be used to perform the cellular works. Even it can be used, this energy can be used for biosynthetic reactions, to make newer molecules, to make more complex molecules. So anabolic pathways, these are metabolic reactions which will consume energy. Again, not every reaction in anabolic pathway has to consume energy, but net result that energy should be utilized to build complicated molecules from simpler compounds. So this is what is example can be photosynthesis, where simple molecules like carbon dioxide, water, they will join to form complex molecules such as glucose. Okay, and the energy which is produced in catabolic reactions, they will be used up in anabolic reactions, giving rise to the hydrolyzed products, that is ADP and PI, right? So as you can see, this is another representation of the same concept. From catabolism, you are getting energy depleted end products, carbon dioxide, water and ammonia. And in anabolism, whatever precursor molecules are there, these monomers, they will undergo polymerization, where they will join with monomers in chains, and they will release out water molecules to polymerize into complex molecules such as proteins, polysaccharides, nucleic acids, etc. In the process of catabolism, there is the release of ATP, NADPH, NADH and FADH2. So these are called as reducing equivalents, okay, because they can carry electrons. Now why are electrons so important? Because they have the capacity to generate ATP. I will explain in oxidative phosphorylation in another video, but ultimately they will lead to the formation of energy. Okay, and in anabolism, these are used up to give back the precursor molecules from which these were built on. For example, ADP and PI, and NAD plus, NADP plus, and FAD. Notice that this is what is called as a PI. It is called as inorganic phosphate. Okay, this is not just regular phosphorus. This can exist in cell independently. That's why it is called as inorganic phosphate. So metabolism is coordinated cellular activity. Sum of all the chemical transformations in organisms, chemical energy, precursor molecule, macromolecule, specialized biomolecule. So overall, just remember that catabolism is a degradative phase, whereas anabolism, it is a biosynthetic phase. Okay. There are three types of metabolic pathways that we see. So some are converging metabolism. For example, from phospholipids we get fatty acids. From glucose also, through the buildup of pyruvate, we will get acetyl-CoA. Fatty acids ultimately lead to acetyl-CoA. From amino acids also we will get acetyl-CoA eventually. So these are converging pathways, catabolism, because complex ones are made into simpler ones. We have diverging anabolism. That means from a simple molecule, more and more complex molecules can be formed through different metabolic pathways. For example, acetyl-CoA is a precursor. Precursor means from which more molecules, complex molecules can be made. It can lead to the formation of eicosanoids also, triacylglycerols also, phospholipids also, and in fact even bile acid steroid molecules through the cholesterol molecule. Okay. And simple pathways are nothing but you can take any one of these, like glycolysis to pyruvate. It will be a simple pathway, and these are more complex pathways. Okay, even cyclic is there. As I said, acetyl-CoA, they will be entering the Krebs cycle, which is a cyclic pathway, but here also it is catabolic. The reason why it is cyclic is because it is the central point of most of the metabolic pathways, and that's why, like how we have ring roads in big cities, right? It is to keep the traffic moving. So from entry points also will be there, exit points also will be there, so that continuously the energy flow should be keep on moving. Coming to ATP, since we are talking of bioenergetics, we should know about the major energy component. Well, it is a nucleotide. You must be aware from my other video on nucleic acid chemistry. You can watch it in Biotechnica channel. You'll get to know a lot of details about that. Well, nucleotide is composed of the ribose sugar and the nitrogenous base, and that is what is nucleoside, which we say. But if phosphate groups are attached with it, then they are called as nucleotides. Okay, so this is the difference when we now with the nucleoside. First of all, this is an adenine, which is a nitrogenous base, and which is joined with the ribose sugar with a glycosidic linkage. It is an N-glycosidic linkage. The ribose sugar will join with a phosphate group. If it has only one phosphate attached with the ribose sugar, then we call it adenosine monophosphate. Mono stands for single. If we have two phosphates attached, we will have diphosphate. If we have three phosphates attached, we will have triphosphate. To know about the bonds present, the bond between the sugar and the phosphate, it is called as phosphoester bond. Please note that this is phosphomonoester bond. Again, attention to detail is very important in CSIR. This is not phosphodiester. Phosphodiester you see in nucleic acids, where one phosphate is joined with two sugars. Here the phosphate is joined with only one sugar. That's why it is called as a phosphomonoester bond. The bond present between two phosphates, it is called as a phosphoanhydride molecule. Phosphoanhydride bond. So as you can see, between this phosphate and this phosphate. By the way, the phosphates are symboled. The first phosphate is alpha, closer to the ribose sugar. The next one is beta, the next one is gamma. Okay. Now phosphoanhydride bonds are very high energy bonds. That's why when they are hydrolyzed, when these will be separated, they will release a lot of energy. Okay. So potentially two phosphodiester bonds can be cleaved as two phosphates are released by hydrolysis from ATP. For example, if you see here ATP, first of all, it's not that just the phosphoanhydride bond, one bond is broken. No, that's not the case. It's not just breaking of a simple bond. There is the requirement of a water molecule also. So that's the hidden secret of chemical reactions. You don't see reaction just breaking of one bond. Okay, so some bond will be made, some bond will be broken. For example, here water, it will dissociate into components H plus and OH minus. So OH minus will get associated here, H plus will get associated here. So that is how you are generating the components. Okay, so this will lead to the inorganic phosphate. It requires this OH minus group attached to it, right? So that's why it's written ATP plus water, which will ultimately break one phosphodiester bond. You will still have two phosphates connected. So it will give rise to ADP plus PI. This is very simple, which we would have already studied. But what is not so simple is the attention to this detail. The free energy change for this reaction is minus 30.5 kilojoule per mole. But if you express it in kilocalorie, it will be minus 7.3 kilocal per mole. So I hope you can understand the level in which you have to prepare for CSIR exam, right? So this is not what is generally taught in master graduation and master's level. For this you require a separate attention, separate level of understanding. Okay, so definitely teachers can help you. You can take help from Biotechnica, so that all the concepts you don't have to prepare. You can definitely get it from reference books. If you study from reference books, you can definitely clear CSI. But the question is, who has the time, right? So you can take guidance from experienced teachers who already know the concept, and they can just deliver it to you in which you can easily understand in one lecture, right? So potentially two phosphodiester bonds can be cleaved as two phosphates are released by hydrolysis from ATP. We have AMP attached with this two other phosphates. So these are phosphodiester bonds. If one is cleaved, then you get AMP with only one phosphate, which is nothing but ADP and PI, right? Then we also have the AMP and P further cleaved phosphodiester bond. So each time you break a phosphodiester bond, even if you cleave this ATP with water, two times if you cleave, then means if you break only one phosphodiester bond, then there is another possibility that you will get PPI. That means two phosphates you can get together, and AMP. So this PPI is called as a pyrophosphate. The PI is only called as an inorganic phosphate, whereas PPI is called as a pyrophosphate. As you can see, the energy is more or less again same, right? And in fact, this PPI can be further broken into PI, because between the two phosphates you have the phosphodiester bond. So this can also be broken and you can give rise to PI, right? So again, here also you have similar amount of free energy released. So this is how we can also have AMP can be broken to release two phosphates also, and two phosphates PPI can be also broken to give two inorganic phosphate. The reason how it happens is because, see, this is the ATP, and as I said, it is hydrolysis. This is the detailed enzymatic reaction, where you can see this is the water molecule. We all know water molecule has a lone pair of electron, which attacks, makes a nucleophilic attack on this third phosphate or the gamma phosphate. Because of this bond, there is the shifting of electrons towards this. So that's why this will carry a negative charge, and part of water molecule, that is H plus, balances this negative charge. So O minus and H plus becomes OH, and the rest of the molecule OH minus gets attached with the phosphate. Okay, so that's where you have the OH group. Now this molecule, as I said, this inorganic phosphate in biological molecule is a stable molecule. The reason because it is having resonance stabilization. So that's why it can exist independently. This is what is our ADP, right? And the free energy for this is minus 30.5 kilojoule per mole. As I said, these are very high energy compounds. So these are represented, phosphoanhydride bonds are represented by this symbol. Compounds with high energy bond are said to have high group transfer potential. So they can actually transfer part of the molecule to another part. For example, we have this PI may be spontaneously cleaved from ATP for transfer to another compound, like as we see in the first reaction of glycolysis, where we have glucose to glucose 6-phosphate. What happens there? A PI is transferred to the glucose OH group, isn't it? So these are phosphoanhydride anhydride bonds which are formed by splitting out water between two phosphoric acids, or even it can be between carboxyl and phosphoric acid. But this particular process will have a large negative free energy change of hydrolysis. If it goes for breaking of the water molecule, breaking of the phosphoanhydride bond, then it will have a large free energy change. Okay, see what reaction they are saying? Hydrolysis. That means they are talking of the hydrolysis reaction, right? Well, there are other high energy compounds in metabolic pathways also. For example, phosphoenol pyruvate is one, 1,3-bisphosphoglycerate and phosphocreatine. If you see phosphoenol pyruvate, which is the second last step in the glycolytic process. So phosphoenol pyruvate, it is converted, in fact the last step, phosphoenol pyruvate, it is converted into pyruvate, which can exist in enol and keto form. That's tautomerism. That's a different issue. But here the enol group which we find in phosphoenol pyruvate, again it is a high energy bond. So that's why when it is broken, again hydrolysis, water molecule is required for that. It leads to the release of a lot of free energy. That is why in the last step, if you remember, in glycolysis, there is the production of one ATP molecule. Okay. In fact, you can see the free energy is so huge. We know for the formation of one ATP molecule, we will need somewhere around 30 kilojoule per mole, isn't it? But as you can see, double of the free energy is released. So here is a fun fact for you, that the energy which is released in the last step of glycolysis, it is enough to generate 2 molecules of ATP, but only 1 ATP is generated. Where is the other energy going? Well, it is released in the form of heat. Okay, so have you noticed that when you are vigorously exercising, lot of glycolysis is taking place in your cell, isn't it? Your body heats up. Okay, so that's the reason the glycolysis last reaction is generating so much heat in your body, right? Then we have the 1,3-bisphosphoglycerate. This here we have the stabilization by ionization of a direct product, which is phosphoglycerate, and the resonance stabilization of the ionized product is seen, just how as we saw in the inorganic phosphate, right? So 1,3-bisphosphoglycerate here will leave out a phosphate group. As I said, pi groups, they can be joined with a metabolic pathway, or it can be left from metabolic pathway. So this also will require hydrolysis. Even this is a high energy reaction. Okay, and then we have the phosphocreatine. So phosphocreatine, and yes, this is also another ATP producing step. Okay, so here also one ATP molecule will be produced, and phosphocreatine, where it is converting into creatine, the resonance stabilization of the product is done, and here also the reaction is spontaneous. It has a negative free energy change. So yes, in CSIR exam, these details are actually important. Okay, these are the details of phosphocreatine, which I already talked about you. So when phosphocreatine is produced when ATP levels will be very high by the enzyme creatine kinase. Okay. Even another high energy compound bond is thioester, which we see in acetyl in the Krebs cycle when pyruvate is converted into acetyl-CoA. So thioester is actually formed between a carboxylic acid, this is the carboxylic acid functional group, and a thiol group. Whenever sulfur is involved, we will call it as a thiol group. So this is a coenzyme, and together they will form coenzyme SH. So co-SH, what we say, that is abbreviated as co-SH, it is the thiol of coenzyme A. Thioesters also have high free energy linkages. So in contrast to phosphate ester, thioesters have a large negative free energy of hydrolysis. So if you see over here also the acetyl-CoA, when it will be attaching with the co-SH, so that's where there is an intermediate after which the acetate molecule will be formed. So when acetyl-CoA loses the CoA, that means it is breaking the thioester bond. Here also the free energy change will be negative. That means it will be a spontaneous reaction. Okay. ATP has special roles in energy coupling and phosphate group transfer. The ATP hydrolysis releases energy which is not able to promote a chemical reaction in an isothermic system, and ATP is involved in many enzymatic reactions. That is, the hydrolysis of ATP adds free energy to the system. The phosphoryl groups are transferred to the reactants or to the amino acid residues belonging to the enzyme, and that's how the free energy will increase. So what you can see as a one-step reaction is sometimes they are having this two-step. Actual reaction will be two-step. That's why whenever you are calculating any energy changes, you should also look at the actual steps, whatever components are getting added or lost from it, right? And coupled reaction are a very important mechanism in metabolism. The exergonic hydrolysis of ATP, which releases a lot of free energy, that's why it is called as exergonic. These reactions will take place along with endergonic reaction. So while one reaction releases energy, the other reaction utilizes that energy in order to take place, right? So this is a process of transferring a phosphate group to another molecule. Basically, one spontaneous process will drive another non-spontaneous process because it cannot take place on its own. So free energy changes of coupled reactions, they are additive. For example, if you have some half catalyzed reactions, okay, so they can be interpretable as two coupled half reactions. One will be spontaneous and other will be non-spontaneous. For example, at the enzyme active site, the coupled reaction is kinetically facilitated while individual half reactions are prevented. Free energy changes of half reactions may be summed up to yield the free energy of the coupled reaction. Take the example of glycolysis enzyme hexokinase. So what we might generally know is that glucose plus ATP gives you glucose 6-phosphate plus PI, but actually it is not like that. One, you have the half reactions, that is ATP hydrolysis. This is one reaction which generates minus 31 kilojoule per mole. Another one is PI plus glucose gives you glucose 6-phosphate plus water. So this, as you can see, this free energy change is positive, which means that this is a non-spontaneous reaction. This is the endergonic reaction, while this is the exergonic reaction, right? So this will not take place on its own, but the ATP hydrolysis releases so much of energy that it can make this reaction take place. But as you can see, it is minus 31, so some energy is utilized. So when this overall reaction is taking place, ultimately you still release free energy. So the net reaction is also exergonic. Okay, so this was what I meant, that the free energy change, free energies of the half reactions, they are additive, but make sure to do it along with sign. You can add up the free energies, but you have to do it along with the sign. Okay, the structure of the enzyme active site from which water is excluded prevents the individual hydrolytic reactions while favoring the coupled reaction. So this is how what I meant, biochemical reactions are coupled. You have the free energy. Glucose is less free energy. Glucose 6-phosphate is high free energy. So this would require input of energy, whereas ATP is high energy, ADP is less energy. So this would release free energy. Some of this free energy is utilized to drive this reaction. Ultimately, you get the complete reaction where also free energy is released. I hope you got an idea of the bioenergetic basic principles. Well, there are a lot of details into it. For example, the different chemical logic and common biochemical reactions. Do let me know in the comment section some types of biochemical reactions. Tell me what are the different type of biochemical reactions that occur in metabolism. Thank you everyone. That's all for now.